last but not least, we have Paul Rathouz, PhD 1997. I have here, and I think in 2010, Paul moved from the University of Chicago to the University of Wisconsin to take the chair and become a professor in the Department of Biostatistics and Medical Informatics. So, Paul, thanks for coming.
So while the slides are coming up, thanks, Mei Chen, for inviting me, and thanks to the department and Mei Chen for organizing it. Thank you, Dr. Rohde, for providing a wonderful environment for students. I will say, because Patrick and I were talking about this letters in our Hopkins file last week. So and he told me this story that I had this exam letter, and it said I could go on, but it didn't actually say that I passed. And so I went back and looked at my letter, and I passed. You might want to double check. I'm going to talk about some science here. Higher order genetic and environmental structure of child adolescent psychopathology. And I'll tell you a little bit why I chose this topic. It's kind of one of my favorite applied papers that I've had the good fortune to participate in. My key collaborator on this is Ben Leahy at University of Chicago, and the reason I wanted to mention him was he's a child psychologist, and one of the things I learned, and other people have alluded to this, is you don't need to be passive biostatistician. You don't need to wait for collaborators to come to you. And so when I interviewed at places, I asked to meet with people outside of the department as an interviewee, so I said, when I interviewed at University of Chicago, I said I want to meet with someone from psychiatry, and so I met with this guy Ben. And we kind of hit it off during the interview process. And then when I got there, I had lunch with him, and we've been collaborating for the last close to 20 years now. And I don't have any conflicts that I know of. So Dr. Rohde's impact and influence on me. So first encounters, when I was a Peace Corps volunteer, when I got back from being a volunteer, I was poking around in public health programs. And I don't know how I set this up, but I came up here. I don't know if you remember this, Dr. Rohde, but I came up here before. I didn't have a master's degree, and I was exploring biostatistics. And I probably knew what a mean was, but probably didn't really know what a variance was. And Dr. Rohde sat down with me, chatted with me for 45 minutes or an hour. And the thing about Dr. Rohde is he always assumes you're a colleague. He always assumes the best in you. You're not a naive student. You're not overly cocky. Even if you are a naive student and overly cocky, none of that. He treated me really like a colleague and told me all about the field and what Hopkins could offer. And then I went off to UNC and did a master's degree. And then a couple of years later, I'm applying, and I come back here. And I sit down with Dr. Rohde, and it was like I saw him 3 days ago. He's like, "Oh, how you doing? Nice to see you again. Let's talk again." And again, he was very welcoming, but always assuming, treating you like a colleague. And then as students, I always felt like we were critical to the department. That there were faculty, staff, students were part of the stakeholders. They were part of the structure. They were part of the mission, and we were always treated like that by Dr. Rohde. So I really want to thank you, Chuck, for setting that environment and making us feel like we were a critical component, echoing what other people have said. And then in terms of the science that I'm going to talk about today, one of the areas that Dr. Rohde was always speaking about was covariance structures, correlation matrices, what you could learn from them, how to interpret them, and linking that to variance components models. And then also, he was also responsible for putting me on psychiatric epidemiology and biostatistics training grant, and then that has colored my career since then. So the talk that I'm going to give is really on an area in behavior genetics using twin studies, but it ties back into these very first decisions that essentially Dr. Rohde made for me because I didn't know enough to make it myself. So thank you for that. Originally, when Mei Chen emailed me, she said, "I want you to talk about leadership, and you have 20 minutes. So then I said, "Okay, I'll do that." And then I got a merciful email about 3 months later saying, "You can talk a little bit about leadership and you can talk some about science." I was like, "Oh, thank God." So here's my one slide on leadership. And these are the things that I just sort of picked up all the way. So, and other people have alluded to these things, but what I try to remember every day is, listen to all the sides, but then, decide how you're going to do something, and take the data into account, sleep on it, and then make a decision and move on. And my sense is there was a lot of mysterious workings from our student perspective about how Dr. Rohde worked, but he always did that. He understood everyone's side, he understood everyone's perspective, so he gets a check on that. The second thing is be authentic, be purposive, be responsible, step up to the plate. And the thing is, if you don't step up to the plate, there's one of two things will happen. No one will, and that's undesirable, or someone else will, and that also can have not the desired effect. So again, whenever anything happened, Dr. Rohde always appeared to be there, took care of things, had the mission of the department as his primary mode of moving forward. The third thing is, and I sort of adhere to this, I think, I don't try to hide my weaknesses. With faculty, I just share, where I feel like I'm doing well and where I feel like I'm not doing well. Now, for Dr. Rohde, that's not applicable because he doesn't have any weaknesses. So we don't have to, he is missing data on that point. Yeah. And then the last thing, and really I think Dr. Rohde exemplifies this, is you have to be present physically and mentally every day in and day out. If you feel lousy, you don't want to go to work that day, that's great for everyone else, but you kind of have to be there, you have to be positive, you have to be optimistic, and it just really makes a difference when you do that, and it's tough to do sometimes. It's not always easy, and 15 years as chair, doing that every day is you value the weekends. But I'll give him a check on that too. So, and these are things that I've sort of trying to do, and then when I look back, I realize that that's what Dr. Rohde did all the time. So, thank you very much for that. So, a little bit about science. The research goals in this project, we notice that a lot of people have noted that, so I'm going to talk about common psychopathology in children. So, by common, I mean depression, anxiety, ADHD, conduct problems, not autism and schizophrenia and things like that. Those are less common, very severe, but less common disorders. And we notice that these dimensions in psychopathology are highly correlated. And we posit that reflects underlying etiologic structure. So we have this hypothesis that a lot of this correlation that we see is due to genetics. And the geneticists, genetic epidemiologists call this pleiotropy, where you have a gene or a set of genes or a collection of genes that impact multiple phenotypes all at the same time. And so we'd like to investigate this hypothesis using behavior genetic analysis of twin data. So I'll skip some of this, but try to go through. For those of you who have never been exposed to twin studies, there's a classic model called the ACE model, and this is how it works. You have some phenotype M that you observe, and you observe it on twin pairs, and you have monozygotic, those are identical twins, and dizygotic fraternal twins. And then you presume that that phenotype is due to three different sources of variability. One is genetic influences, that's A. And so what we're going to, in this setup, the capital letters are normal random variables, variance 1, and the little letters are coefficients. And then you have things that are in the environment that vary from family to family, but that are shared with all siblings in a family. And then you have E, which are things that are in the that are specific to that individual. And now people say, well, I'm assuming that these are additive, but actually they're not really. It's not really so much as an assumption, but it's almost like a definition. In other words, A is everything that is linked to genes in any way, causal or not, and then C is everything that varies from family to family that's not linked to genes, that's not correlated with genes, and then E is everything else. So it's almost like the assumption is satisfied because it's defined like that. And then we get this critical notion called heritability, which is the variance of the in the denominator is the total variance, and then the numerator is the variance due to genetic factors. And that is really, that heritability notion, even though it goes way back to classic twin studies from the 60s and 70s, has been carried forward into modern days of molecular and genetic epidemiology. And so these designs permit identification of this model. And the reason it works is that we assume that the genetic influences on two identical twins are perfectly correlated, and the genetic influences on two fraternal twins are only correlated at an average rate of 1/2. And so that differing genetic relationship leads to model identification. That's basically a slide that just says that, so I'll skip that part. Now, the nice thing about this, or the kind of really interesting thing about this, is when you carry this out into a multivariate setting. So suppose instead of a single phenotype, I have two phenotypes or more phenotypes. Now, everything that's a variance becomes a variance-covariance matrix, and I have a variance-covariance matrix of genetic influences, a variance-covariance matrix of environmental influences, and so on. And those shared genetic influences are of great scientific interest. And so this direct multivariate extension of the twin study allows us to look into those. So now this field, twin studies, and this is where the courage piece comes in. This is a kind of a little cottage industry of psychology. You think Hopkins as a family. This sort of a behavior genetic group they're really a family. And there's grandparents and there's a whole situation in there. So getting into this field can be a little daunting. It's a difficult area to work in for that reason, but it's an important area because this heritability construct, as I said, has been carried forward into these modern eras, and it essentially provides an upper bound on what we can always expect to learn from any genetic study. And so we're going to use it in this study to sort of examine the taxonomy of childhood psychiatric disorders. So in this study, we had about 1,500 twin pairs, which is a phenomenally large twin study, with a couple of exceptions of registries. They were all 9 to 17 years old. They were born in Tennessee, and it was a roughly, approximately representative sample. If you work in twin studies, you see that you get about 1/3 monozygotic twins, about 1/3 same-sex dizygotics, and about 1/3 different-sex dizygotics. And we looked at 11 prevalent childhood psychiatric disorders. And so we know that they're going to be highly comorbid, and we're really interested in how much is that comorbidity due to genetic factors. So here's a little model. This is our ACE model on the right, and on the left is an 11 vector. I didn't list them all there, but we have major depressive disorder, generalized anxiety disorder, and the one on the bottom is hyperactivity and inattention. And we assume that each of those dimensions has an A component, a C component, and an E component. And it turns out in many of these, C is kind of small, so I'll just kind of ignore that for right now. So it kind of boils down to an A and an E component, broadly construed. And so a first goal is to estimate the variance of A and the variance of E, and that's a quantification of the genetic environmental and environmental influences. So we did that, and this is a table, and in green, in the vertical green box, I have the variance due to genes for these 11 conditions, and in the horizontal green box, I have the variance due to environment. And you can see that there's a lot of variance due to genetic factors in here, 61%, 50%, 78, 79%, and so on, but also a substantial amount of variance due to environment. So the next question we asked, and I think I got these slightly out of, then the next question, then what's in the boxes here, on the upper diagonal are the covariances due to environmental factors. And so these are all the covariances amongst these, and this is the beauty of these twin studies, is it allows us to decompose this environmental and this genetic piece. So these are all the covariances due to environmental factors, and you can see that there's some strong ones in there. There's a 76, but otherwise they're all well under 0.5, and some of them are really quite small. So this suggests sort of a weak environmental structure linking these 11 prevalent dimensions of psychopathology. Below the diagonal are the genetic correlations, and this is why I love this paper, because this result is so striking right there in the correlation matrix. The correlations here are quite substantial among the 11 dimensions of psychopathology, and truly suggest that there's an enormous amount of genetic overlap across these dimensions. The same genes are impacting a variety of different dimensions. So the next question we asked then, so the summary so far is far and away the comorbidity that we observe phenotypically among these disorders is accounted for by shared genes and shared genetic factors versus shared environmental factors. We went on to sort of ask, here's my model again, we went on to ask, can we impose a structure on this? And so we built a hierarchical model where we also then have another sort of a latent variable model underneath it. Again, latent variable models are something we explored when I was a student here at Hopkins. And so we posited 3 underlying factors, a general factor, an internalizing factor representing things like depression and anxiety, and an externalizing factor representing more behavioral phenotypes like hyperactivity or conduct problems. And then we did that for both the environment and the genes. And then this is the result for the genetic effects, and this is a sort of box and arrow diagram. The circles are unobserved latent quantities, and the boxes are observed quantities. So what we can see here is for the 11 factors, there's an enormous amount of influence from this general factor, G, And then there's an internalizing and externalizing factors that are also pretty influential. And then the numbers at the bottom represent kind of the unique genetic influences that only impact one phenotype at a time. And by and large, some of those are quite small. Conduct problems being on the left being the biggest one, but many of them are quite small. Environment's a little bit of a different story. There's a lot of individual variance that's specific to each of those 11 factors. You can see the numbers on the bottom, 0.64, 0.56. That represents the contributions from environmental shocks that only affect a single phenotype. And so the summary and implications are this sort of notion of generalist genes. There's a general genetic predisposition. We have a high amount of pleiotropy. Single genes or collections of genes affect multiple phenotypes. And that are globally associated with all these different dimensions of psychopathology. On the other hand, we have specialist environments. Different shocks that you get in the environment lead to different phenotypes. This has really profound implications for genetic epidemiology, because if you want to do genetic studies and you just focus narrowly on a single phenotype, you could be really hurting yourself in terms of power, because you're throwing away a lot of you're essentially misclassifying a bunch of individuals as not affected. And now, if you read grant applications in this area, genetic epidemiology, especially psychiatric epidemiology, pleiotropy is a regular design concern. So it's if you read the grant application, it's right there with how are we going to deal with confounding population substructure, missing data, how are we dealing with pleiotropy. It's a factor that is highlighted. And that's what I have to say. And so I think but I do think this project kind of illustrates many things that have come together for me. That I learned starting here when I was a student at Hopkins. So thank you, Dr. Rohde, for setting the context for that and being on my dissertation committee and passing me in my exams and things.
Okay, so I'm sorry, I can't help but make one comment. I think Paul just rolled out a new definition. So in statistical genetics, when you do big family studies, there's a well-defined notion of alleles being identical by descent. I think Paul just introduced statistical models being identical by descent in large families. This is brilliant. So Mei Cheng asked me before we call it a session to point out to you there's a refreshment station out there. If you go out there, take a left and look towards your right, there's a big screen we call it the Wall of Wonder, and there should be coffee carts and water and things like that. So with that, hope you all join me in thanking our speakers again.